Hello. The cell is the basic unit of all living organisms. All functions or abilities of animals can be traced down to the cell. Sometimes, by looking at a highly magnified image of a cell and some basic knowledge of the functional parts of a cell, you can already guess what the specialized function of that cell is. Just like the image of the cell that is shown here at the backdrop of this introductory slide. The watermark image is a plasma cell. By function, the plasma cell is an activated white blood cell that acts as a factory of antibodies. Antibodies are very tiny, Y-shaped molecular proteins that act like the police handcuff. They bind to disease-causing invaders in our bodies to keep the pathogen from harming our cells. The plasma cell shown here shows a very extensive membranous organelle that is most likely an endoplasmic reticulum, particularly the rough endoplasmic reticulum, because it's dotted with very tiny particles that is most likely ribosome. The rough endoplasmic reticulum is responsible for synthesizing proteins that are for export by the cell. This extensive rough endoplasmic reticulum that occupies the cell's entire cytoplasm here must be very busy manufacturing antibodies that fight invading pathogens. And so, it is very important then that understanding the cells better and to be able to know better the workings of an animal's body, a good working knowledge about the cell is necessary. At the end of this discussion, you should be able to describe the cell concept, enumerate the methods of cellular studies, identify the cell structures and functions, describe the plasma membrane structure and functions according to the fluid mosaic model, and differentiate the various membrane transport mechanisms. The last remaining subtopics will be your task of reading and understanding. In your task on mitosis and cell division, you must be able to characterize the events of each mitotic phase and describe the stages of the cell cycle and its regulation. What do we know about the cell so far? Cells are incredibly small but are very complex units of living organisms that up to this point in time, it's still far from being completely understood. There would be no cancer anymore today if we did. The unified cell theory states that all living things are composed of one or more cells and their products. The cell is the basic unit of life, and that new cells arise from pre-existing cells. The expanded version of the cell theory also includes the declaration of cells that they carry genetic material that is passed on to daughter cells during cell division. And all cells are essentially the same in chemical composition, and that energy flow, according to metabolism and biochemistry, occurs within cells. In single-celled organisms, all life functions are confined to that one cell. But in multicellular organisms, the cells specialize and interact with each other in an organized partnership, like a corporation. Cells have to be minuscule in order to maintain high surface to volume ratio. But why is this? I'd like you I'd like to leave you this question to think about this. Why can't the cell grow any bigger? Why does it have to be small? I'd like you to add your answer to this question to the assignment that I will give later. The figures that are shown to the right of this slide are the major contributors to the cell theory in its beginnings. You have Robert Hooke, who in 1665 published his book Micrographia, in which he first coined the term cell, when he described the repetitive units of cork tissue. Anton van Leeuwenhoek, a microscope hobbyist, submitted his letters to the Royal Society journal that published his microscopic findings of cells, including bacteria. For this, he is largely recognized as the father of microbiology. But it was about 200 years later, in the 1830s, that Matthias Schleiden and Theodor Schwann published their idea that all plants and animal tissues are made up of cells. It was only that time that the cellular theory was recognized as acceptable. In 1839, Johannes Perkenyi coined the term protoplasm as the fluid substance of the cell. And in 1855, Rudolf Virchow published his cellular theory that all cells come from pre-existing cells. These six persons are the giants on which many of the cellular biologists sits on in their ideas. There never has been any biological concept and entity that is as intimately associated to a device than the cell and the microscope. Much of the initial understanding of cells may be ascribed to the development and sophistication of the microscope. But of course, alongside the microscope were the more creative biochemical and molecular techniques that has also advanced many of the functional understanding of cell organelles by basic staining, isolation, fractionation, and many more molecular techniques. The advent of the powerful electron microscopes broke through the limits of light-based optical studies by producing high-definition and color-enhanced subcellular imageries, which supplemented more advanced molecular techniques of cell and molecular biology. New techniques of molecular labeling, staining has also advanced light microscopy today, such as visualization of the locations of specific genes, RNA transcripts, or proteins is already routinary in research. Let's just briefly visit some of the images here that's shown in this slide. The paddle-like device that's shown here was Anton van Leeuwenhoek's ancient microscope. He had to peer through a very tiny lens at the specimen at the back of this board so that he could see the microscopic images. Anton van Leeuwenhoek was actually a merchant, but his hobby was making lenses. And because of his notes and the discoveries about tiny animalcules that he saw under his microscope, he was able to initiate the beginnings of microbiology. And here you have the microscope of Robert Hooke, who was a professional scientist, a biologist who peered through many tissues, and one of the tissues that he studied was the cork tissue, and he was the one who first stated that who coined rather the word cell when he looked at cork cells that were made of tiny little rooms. And here you have the microscope of Matthias Schleiden that's similar to Theodor Schwann's microscope, and because of these microscopes they were able to formulate the first statements of the cell theory, and that is that animal and plant tissues are all made up of cells. And at the center of this image, rather of this slide here, are the diagrams of the light microscope on the left and electron microscopes on to the right and the center column. You have the transmission electron microscope that's diagram here, and onto the right you have the scanning electron microscopes. Now the main difference between these microscopes, light and electron microscope, is the illuminating medium that is used and the magnifying device that is used. In light microscope you have the ordinary light, the visible light that is used, and the device to manipulate and control the passage of light in order to magnify an image are glass lenses, mostly convex glass lenses. And then in the electron microscopes, the illuminating medium is a beam of electron that is passed along points with magnets, and because electrons are negatively charged. If you have magnets that is oppositely charged, you can bend the beam of electrons. And just like in light, when glass lenses bend the light, the beams of electrons can be bent as well so that it could magnify the image of a very, very tiny subcellular structure. Let's move on. Cells are basically differentiated as either prokaryotic or eukaryotic cells. Prokaryotic cells are thought to be more primitive and simpler cell types, characterizing bacterial cells or microorganisms. They demonstrate the lack of nucleus, hence they are called prokaryote, meaning cells before the appearance of cells with nucleus. Prokaryotes, which are about 1 to 10 micrometers in size, are much smaller than eukaryotic cells, which are about 10 to 100 micrometer. The chromosomes of prokaryotes are circular and are not membrane enclosed, but confined to a region in the cytoplasm called nucleoid. While that of the eukaryotes, the chromosomes are linear and are heavily bound to globular proteins, which are enclosed in double-layered nuclear membrane. And so you have a distinct nucleus in eukaryotes because of these nuclear membranes. The prokaryotes replicate mainly by binary fission, while eukaryotes replicate by mitosis. There are many more differences of prokaryote and eukaryote, but we have to move on right now. I'll just give you some more materials, or you can refer to the textbook to look at other differences between prokaryote and eukaryotic cells. The eukaryotic cell is highly compartmentalized. Compartmentalization makes animal cells highly complex and efficient because with many different spaces, the cell can carry out many functions that are otherwise incompatible if they were to happen in just one space. With compartmentalization, the cell can also carry out processes in progressive stages, a feature of highly organized living entities. There are three basic parts or regions of a cell that is outright obvious when you look at a cell under light microscope. This includes the nucleus, the region around it is the cytoplasm, and then the boundary of the cell formed by the plasma membrane. In certain nuclei of certain cells, the nucleus can also manifest a small ovoid densely staining region called the nucleolus. The nucleolus is actually a cluster of active DNA and RNA and proteins that are engaged in synthesis of messenger RNA and ribosomal subunits that are shuttled out of the nucleus later for protein synthesis. The nucleus under electron microscopy can also show linear thin strands of chromatin, the unwound form of chromosome in a cell that is not engaged in cell division. The nuclear membrane under electron microscopy is also shown to be made up of two membranes. So you have double membrane nucleus, which fuse at certain points to form the nuclear pore. The nuclear pore is formed by proteins that guards the exit and entry of chemicals or substances in and out of the nucleus. So the nucleus is heavily guarded. It is like the Malacanang or White House of the cell, which governs the activities of the cell throughout. Outside the nucleus, you will also find several kinds of membranous organelles. This includes the endoplasmic reticula, composed of two, the smooth endoplasmic reticulum, which is formed by a network of anastomosing membranous tubules, and then you have the rough endoplasmic reticulum, made up of membranous sacs that are studded with ribosomes. The smooth endoplasmic reticulum is responsible for lipid synthesis, for the metabolism of carbohydrates, for storing calcium in some cells like muscle cells, and for detoxifying drugs and poisonous substances. The rough endoplasmic reticulum, on the other hand, is responsible for synthesizing proteins that are for self-secretion or export. Apical to the endoplasmic reticulum is a stack of membranous sacs forming the Golgi body. The Golgi body is known to sort and modify cell products like adding sugar moieties in glycosylation, and these products are then packaged in small membranous vesicles. The vesicles themselves are cellular organelles for transport of cell products to their final destination. Both the nuclear envelope, the endoplasmic reticula, and the Golgi apparatus and vesicles form the endomembrane system of a eukaryotic cell that regulates the traffic and formation of cell products. Other membranous organelles may also be found here in this illustrated generalized cell. You have mitochondria that are cut cross-sectionally to show the internal structures. The mitochondrion is like the nucleus, which is double membrane. The inner mitochondrial membrane is heavily folded inwards, forming cristae. The mitochondrion has many enzymes here that synthesizes ATP, or adenosine triphosphate, using the energy coming from the electrons shuttled from the metabolism of carbohydrates, lipids, and proteins. So the mitochondrion is the region or the organelle where the final stage of cell respiration occurs, which results to the synthesis of ATP. Because of this, the mitochondrion is often dubbed as the powerhouse of the cell. The mitochondrion that is shown here are elongated or sausage-shaped mitochondria, but in many cases the mitochondrion is actually oval or round shape. There are also other single membrane organelles here like the lysosome. The lysosome contains corrosive enzymes that can break down food molecules. When lysosomes bind to food molecules, they form phagosomes. The lysosomes are also used to break down cell structures that are needed for recycling or renovation. In white blood cells, the lysosomes bind with ingested bacterial cells to destroy the bacteria with lysosomal enzymes. There's another single membrane organelle here that contains enzymes, the peroxisome. Peroxisomes under electron microscopy is shown to have densely staining region, and these peroxisomes contain enzymes that can break down free radicals or poisonous substances and convert them to hydrogen peroxide that immediately breaks down into water, rendering the toxic products into harmless water. Other non-membranous, the plasma membrane is another organelle, a multifunctional organelle of the eukaryotic cell. The plasma membrane serves to transport substances in and out of the cell, a topic which we will discuss also in this lecture. The plasma membrane can aid in cell-to-cell recognition, such as those done by immune cells when they do surveillance of the body. They have to recognize which is a cell that is part of the body and which is a foreign cell by cell-to-cell recognition. The plasma membrane can also bind with extracellular chemical signals and transmit the signals interiorly by initiating a cascade of chemical reactions that ultimately lead to the cell's response. We call this a signal transduction. The plasma membrane can also form specialized junctions between with adjacent cells, forming intercellular junctions. There are also enzymes in the plasma membrane formed by uh, proteins that participate in metabolism. So the plasma membrane can also uh, serve in metabolism. Okay, in this illustration, the plasma membrane is shown to possess short finger-like projections called microvilli. This is characteristic of many intestinal cells. The microvilli can elongate or shorten depending on the action of the core of microfilaments inside. What the microvilli does generally is to increase the surface area of the cell when it is about to absorb nutrients or other substances. The increase of surface area increases in turn the amounts of nutrients or substances that can be assimilated by the plasma membrane. There's also other organelles that are shown here that are not membrane based. This includes the centrosome, which is formed by a core of short barrel-shaped microtubule array called the centrioles. The centrosome is the microtubule organizing center of the cell. When needed, the centrosome can produce microtubules or assemble microtubules, such as during cell division when the chromosomes are shuttled to the opposite sides of the cell. This is served by forming microtubules or spindle fibers by the centrosome. Besides microtubules, the cell also possesses smaller strands of protein fibers like microfilaments and intermediate filaments. Collectively, these microtubules, microfilaments, and intermediate filaments form a network of crisscrossing fibers throughout the cytoplasm. We call this as the cytoskeleton. The cytoskeleton supports the cell structure or the cell shape. In certain cells that undergo change of shape because they have to crawl over surfaces like the amoeboid immune cells, these cytoskeletal elements or cytoskeleton usually supports this change of shape as well by the reassembly and disassembly of cytoskeletal elements. The cytoskeleton can also anchor certain organelles at specific regions in the cytoplasm. The cytoskeleton can also serve in guiding the transport of substances or cell products throughout the cell cytoplasm. So the cytoskeleton formed by the microtubules, intermediate filaments, and microfilaments is a very crucial scaffold of fibers that has many functions as well. There's also an extracellular locomotory organelle that is shown here in this generalized cell illustration. This is the flagellum, which is made up of a core of microtubules that are basally powered by mitochondria, and the whole length of the flagellum is made up of, or rather is covered by plasma membrane. This flagellum is a locomotory structure that executes a whip-like motion that propels cells. For example, sperm cells are propelled by the undulation of flagellum. So there you see the cell structure and organelles can be subdivided into membrane-based organelles that includes the nucleus, the endoplasmic reticula, the Golgi apparatus, mitochondria, lysosomes, and peroxisomes. There are also cell organelles that are non-membrane based, like the centrosome, the cytoskeletal elements, and as well as the ribosomes, the free ribosomes that are suspended in the cytoplasm. The free ribosomes are separate from those ribosomes embedded in the rough endoplasmic reticulum. They are freely suspended in the cytosol, and when they synthesize proteins, these proteins that are produced by free ribosomes are for cell consumption, proteins that are for the use of the cell itself inside. Those proteins that are synthesized by ribosomes embedded in rough endoplasmic reticula are for cell secretion to be used outside the cell. Now, the fluid mosaic model is the currently accepted concept describing the plasma membrane structure. By electron microscopy, a plasma membrane appears as two dark lines, each approximately 3 nanometers thick at each side of the light zone. The entire membrane is 8 to 10 nanometers thick. In this larger artist's depiction of the plasma membrane, the phospholipid bilayer forms the main structure of the plasma membrane, with proteins variably inserted into them. Some are peripheral, mostly located at the intracellular side, and a number of the proteins are transmembrane proteins or integral proteins that insert themselves the whole depth of the lipid bilayer. In the lipid bilayer, the phospholipid heads are oriented towards the watery environments of the cell, whether extracellular or intracellular environments. So that's the phospholipid heads there, and the fatty acid tails of the phospholipids face each other to form a hydrophobic zone. Now, because of this hydrophobic zone, the plasma membrane is therefore impervious to small polar molecules, and the lipid bilayer is defaulted to accept or to allow passage only of small non-polar molecules, as the middle layer of the lipid bilayer is made up of non-polar zone. According to the fluid mosaic model, the plasma membrane is held together by hydrophobic interactions, and this is a weak bond between molecules, so that the molecules can actually be very flexible. The lipids can move about in their point of or plane of positions, like partygoers elbowing their way across a filled room. No? And occasionally the phospholipids can flip-flop to the other plane of the lipid bilayer. The outer surface of the plasma membrane is occupied by branching chains of sugar molecules or carbohydrates that are attached to either phospholipid heads or the transmembrane proteins. These carbohydrate chains are very important in that many of these are actually receptors to specific chemical signals. The ABO blood grouping is largely based on this um, carbohydrate chains on the surface of red blood cells that can provide for identification of the blood type. Apart from this short branching chain of carbohydrates that can act as receptors or as determinants of blood type, there are also a lot of proteins and polysaccharides that are enmeshed on the surface of the plasma membrane, forming the extracellular matrix. So the plasma membrane on the surface is not a very uh, void surface, an empty surface, but it's actually very bushy because of the preponderance of extracellular matrix components, especially proteins and polysaccharides. At the inner side of the plasma membrane, there are also proteins that are anchored to cytoskeletal elements that keeps them within those regions of the plasma membrane. The dynamic disassembly and reassembly, however, of these cytoskeletal elements can allow the transmembrane proteins to move around at the plasma membrane in their plane of existence. In most multicellular organisms, the cells are joined together by intercellular junctions. This, if atoms are bound together to form molecules by chemical bonds, in multicellular organisms or in tissues, the cells are bound together by cellular bonds, and these are the intercellular junctions. You have several types of uh, intercellular junctions. You have tight junction that is shown here that surrounds the whole cell at the upper surfaces. These are membranes of two adjacent cells that are fused together. Their transmembrane proteins form as one, and they form rows of transmembrane proteins that are bound tightly between adjacent cells, closing the gap between neighboring cells. And this seals off any substance or liquids that is out at the uh, outer surface or the apical surface of the cell and at the bottom surface of the cell. So it prevents molecules from passing through the intercellular spaces of the cells. You also have uh, adhesion molecules or adhesion junctions rather, which occur just beneath the tight junctions. These anchoring junctions are similar to tight junctions in that they encircle the cell. They are different, however, from tight junctions in that they do not seal adjacent cells, and so there's a passageway for substances to move through along the intercellular spaces between adjacent cells. Transmembrane proteins link together a small intercellular space. Now, you also have desmosomes. Desmosomes are small ellipsoid discs that occur within the plasma membrane of two adjacent cells. Desmosomes act like spot welds between adjacent cells. They hold the cells together. And then you have gap junctions, which provide a means for intercellular communication. Okay, in the gap junctions, the two adjacent or two opposing membranes are brought together by transmembrane proteins. So this region of the plasma membranes of adjacent cells are actually joined together. and you have transmembrane proteins that form channels across the two membranes of adjacent cells, allowing communication, direct communication between cytoplasm of one cell to that of the other cell. That's gap junctions. They form tiny canals between cells so that the cells can communicate continuously. Molecules and ion can pass from one cell to another through these transmembrane proteins in the gap junctions. They are very common between epithelial cells, nervous tissues, and muscle tissues. Hemidesmosomes, that's shown here at the bottom, occur at the base of cells, of epithelial cells, and anchor them to the underlying connective tissue layers. So in this way, the epithelial cells cannot just be sloughed off or um, torn away from the underlying tissues of epithelial tissues because of these hemidesmosomes. Due to the diversity of transmembrane proteins, the plasma membrane is a multifunctional organelle. It serves many functions. As was already mentioned previously, the plasma membrane forms special structures that join cells together or to their substrates. Being the boundary of animal cells between exterior and interior, plasma membrane possesses mechanisms that move substances across the plasma membrane. Many transmembrane proteins also contains enzymes that initiate and participate in metabolism of cellular substrates. Transmembrane proteins can also be crucial to cell-to-cell recognition during embryogenesis, immune surveillance, and immune cell interactions. And lastly, the transmembrane proteins are also important to the binding of extracellular messengers and convert and transmit this external signal towards the cell's interior network that leads to the cell's responses. And so you see, transmembrane proteins and the plasma membrane as a whole has many functions. It is not just about intercellular junctions, anchorage, or transport, but they can also participate in enzymatic or metabolic reactions. They can also participate in cell-to-cell recognition between cells, especially among white blood cells, as well as in the reception of chemical signals from the cell's exterior. Transport mechanisms across membrane is important to cell life. By chemical and structural nature, the lipid bilayer of the cell membranes presents a barrier to many substances, especially polar and large molecules. It has therefore adopted several mechanisms that allows different important cellular commodities to move in and out of cellular locations unencumbered. These mechanisms involve simple physical processes that are common in nature, as well as metabolic energy-driven mechanisms that involve special transport proteins. This slide here, or this section, covers the following: diffusion, osmosis, carrier-mediated transport, and there are four of these: facilitated diffusion, active transport, endocytosis, and exocytosis. The passive membrane transport of solutes across a membrane occurs in two ways: by simple diffusion or by facilitated diffusion. Simple diffusion involves the passive passage of small hydrophobic or nonpolar solutes across the plasma membrane from an area of high concentration to an area of low concentration. The solutes move by their own kinetic energy, and the rate of movement is dependent on the steepness of the concentration gradient across the membrane. When the concentration gradient decreases as more and more solutes move across the membrane, simple diffusion slows down until it consequently stops. This means that equilibrium has been achieved. At solute equilibrium, it does not mean, however, that no solutes are diffusing anymore. Rather, that the movement or diffusion of the solutes between two sides are now equal. Studies have shown that certain solutes, especially hydrophilic, larger solutes, can move across the membranes at a faster rate than what can be accounted for by simple diffusion. And this has been known as facilitated diffusion. Facilitated diffusion is similar to that of simple diffusion because the movement of the solutes is along the concentration gradient, that is, and that it is driven by kinetic energy of the solutes. But two things, or at least three things, differentiate facilitated diffusion from simple diffusion. One is that the movement of the solutes by facilitated diffusion is faster than in simple diffusion. Secondly, it involves a transmembrane protein that facilitates the movement of the solutes unencumbered. And thirdly, because there are limited number of transmembrane proteins in the plasma membrane, facilitated diffusion can reach saturation point, wherein all the transmembrane proteins or carriers are fully occupied by the solutes. At that point, no additional amount of solutes can increase the flux or diffusion of the solutes. To summarize facilitated transport, facilitated diffusion is kinetic energy driven, and it depends on two factors. One is carrier specificity, and second is saturation point. In carrier specificity, it is shown here by this GIF image. When the solute shape matches that of the entry point or the binding site of the carrier or channel, it successfully moves through the membrane, like here. But if the solute does not match the entry point or the binding site of the carrier or the channel, it will never successfully come across the membrane. Secondly, when solutes are moving across the carrier, there is a point in time when all the carriers are fully bound with the right solutes. At that point, all the carriers are said to be saturated. So there's a point in time, therefore, that no additional solutes will ever be able to cross the membrane because all the carriers are fully occupied by the solutes. And that is what we mean by saturation point. The transport proteins in facilitated transport mechanisms exist in two forms, as carrier, as is shown here, and channels. Carriers are distinguished from channels in that they undergo a change of shape or conformational change whenever they are bound with the right solute. In this case of our image, a sugar solute binds correctly with the channel, and the carrier undergoes conformational change and successfully allows the solute to cross the membrane. Channels, on the other hand, have an unchanging conduit or passageway for the solutes that they allow to move across. But channels come in two forms. One, when they are open 24/7, so ions can move or solutes can move across at any time. And there are also gated channels, wherein the channels are opened only when the correct solutes are bound to it or when there is a correct stimulus that opens it up. Otherwise, it stays closed. Gated channels also exist in two forms, either as chemically gated or voltage gated channels. Chemically gated channels open only when a specific ligand or chemical substance binds to it. And when it does, it opens up and allows the substance to move across the membrane. Voltage-gated channels open only when the voltage across the membrane, as shown here by the positive and negative charge at the two sides of the membrane, changes. And when that happens, the channel opens up, allowing the solutes to move across the membrane. This is very common among many nerve cells and muscle cells, that opens only when there is either the correct ligand, like a neurotransmitter, or an alteration of their membrane potential, or the charge across the membrane, that opens up voltage-gated ion channels. Osmosis is the simple diffusion of water across a semi-permeable or selectively permeable membrane. If water is separated by a semi-permeable membrane, its net direction of diffusion is directly affected by the concentration gradients of free water molecules. If there are solutes present in one compartment, while there is none on the other compartment, this creates a concentration gradient of free water molecules. That compartment with solutes will tend to have less free water because some of its water molecules are bound to the solutes, whereas the other compartment with no solutes have more free water molecules. And water will therefore diffuse from an area of high free water concentration to a compartment with less free water molecules. Because this is simple diffusion of water, this process is also subject to equilibrium. When the free water concentrations are more or less equal, the diffusion of water stops, and the level of water therefore is maintained at such level. In the body, osmosis never reaches equilibrium because the cells in animal organs, like in the kidney, actively establish solute concentration gradients so that water can continually move from blood to the kidneys. The plasma membranes of animal cells have special water channel proteins called aquaporins. So depending on the presence of aquaporins and the concentration gradient of free water molecules in and out of the cell, water has the potential to diffuse across the cell's membranes. We've learned, however, in the previous slide, that the concentration of solutes have an indirect effect on free water concentrations across the cell membrane. Solutions with varying solute concentrations relative to the cell's solute concentration are classified as either hypotonic, those with less solutes than the cell, hypertonic, having more solutes than the cell, or isotonic, having solutes equal to those in the cell. Due to osmosis, the following figures of red blood cells shows their fate when immersed in varying concentrations of solutes, that is hypotonic, hypertonic, or isotonic solutions. In the series of red blood cell figures shown in the slide, which do you think is the blood serum osmolarity that is hypotonic, isotonic, and hypertonic to the red blood cells? These cells here were immersed in 200 milliosmoles, showing a bloated shape of red blood cells. But when they were immersed further in 100 milliosmol, the red blood cells sort of lost their contents, and therefore leaving behind ghost red blood cells. Only the cell membranes are left. On the other side, you have immersion of red blood cells in 400 milliosmoles and 500 milliosmoles. Here you see the cells have collapsed, losing their contents to the serum around them. And in the 500 milliosmol, there's so much collapse of red blood cells that their surfaces appear spiky. They seem to have shriveled. Whereas at the middle, with 300 milliosmol, the red blood cell normal shape is retained. The red blood cells are normally biconcave in shape. That is shown here by some of the cells, biconcave appearance. So, which of these solutions, therefore, or sera, are hypertonic, isotonic, and hypotonic? It appears here that the serum or solution with 300 milliosmol is isotonic to red blood cells and perhaps to many human body cells, 300 milliosmol, so that if you lessen or increase that osmolarity, you can infer that it will cause osmotic flow of water in or out of the cell. Active transport always involves an expenditure of energy from ATP because substances are transported against their concentration gradient. The transmembrane protein involved in active transport are complex with an enzyme ATP hydrolase phosphatase that facilitates the hydrolysis and phosphorylation of ATP and ADP respectively. Among the most important active transport systems in animals are those that maintain sodium and potassium ion gradients between cells and its surrounding extracellular environment. Sodium-potassium pumps, as they are also known, are very important transmembrane proteins in nerve and muscle cells that maintains the cell's irritability. Most animal cells require a high internal concentration of potassium ions for protein synthesis at the ribosome and for certain enzymatic functions. There are two kinds of active transport systems, the primary and secondary active transport systems. In primary active transport, the movement of the solutes is powered directly by the hydrolysis of ATP. Okay? And thereby the solutes are concentrated on one side. In secondary active transport, the movement of the solute is powered by the concentration gradient that was established by primary active transport. So it indirectly utilizes the energy from ATP by using the concentration gradient of the solute established by primary active transport. That's secondary active transport. Whole or large substances may be transported across the membranes and into the cells or out of the cells. This requires different ATP-dependent mechanisms that involve whole patches of plasma membrane to assimilate or secrete macromolecules. This includes or involves endocytosis and exocytosis. Endocytosis, the ingestion of materials by cells, is a collective term that describes similar processes of phagocytosis, pinocytosis, and receptor-mediated endocytosis. They are pathways for specifically internalizing solid particles, small molecules and ions, and macromolecules respectively. All require energy and thus are forms of active transport. During phagocytosis, for example, an area of the plasma membrane coated externally with specific receptors and internally with actin and actin-binding proteins forms a pocket that engulfs the solid material. The membrane-enclosed vesicle, a food vacuole or phagosome now, then detaches from the cell surface and moves into the cytoplasm where it fuses with lysosomes. Its content are then digested by the lysosomal enzymes. In pinocytosis, similar to a process to phagocytosis, except that small areas of the surface of the membrane are infolded into the cells, forming tiny vesicles. These infoldings, called pits and vesicles, are called cavulae. The receptor-mediated endocytosis is a specific mechanism for bringing large molecules within the cell. The infolding plasma membrane are coated with protein receptor-rich layer such as clathrin that aids in binding target molecules for uptake. Membrane vesicles within a cell can also fuse with plasma membrane and extrude its contents to the surrounding medium. This is the process of exocytosis, as shown here. Exocytosis serves to remove undigestible residues of substances brought in by endocytosis, or to secrete substances that are such as hormones, or to recycle membrane receptors and membranes, and to transport substances completely across the cellular barrier by transcytosis. So in summary, our understanding of the cell and its importance to life has taken a long 300 plus years of history of discoveries. This understanding has shown us that animal cells are distinctly complex with characteristic components that serve in specific distinct function. The plasma membrane is crucial to uptake or release of life-sustaining substances, employing mechanisms that are kinetic energy driven or metabolic energy driven. As for the topic of cell division, you are assigned to work on the main aspects of cell division by answering the following questions at the next slide. For you to answer, you have these following questions. Why can't cells grow beyond 100 micrometer? In maintaining their microscopic minuscule size, is surface to ratio increased or decreased? Explain your answer. Second question. What events characterize the different phases of mitosis, from prophase to metaphase? What are the characterizing events of the cell cycle, and how is it regulated? And then lastly, since some human cells do not divide anymore, like many neurons or brain cells, if so, what stages of the cell cycle do the neurons or brain cells exhibit? Thank you and good day.